Hello, this is Dr. Michelle Chirinos, and I'll talk to you about an in silico analysis of MSH orthologous protein of major agronomic crops. All living organisms are continuously exposed to DNA damage by different factors, as you can see in this slide. To avoid this change in DNA become mutations that can affect regular cellular processes, exist DNA repair systems. It can be direct or indirect. Indirect DNA repair systems can be BER, base excision repair, NER, nucleotide excision repair, or the mismatch repair, MMR. We will focus in mismatch repair systems because there are few studies about that, especially in plants. For example, this is the first time that someone models mismatch repair proteins in rice, soybean, and corn. This work aims to characterize in silico the MSH proteins of major agronomic crops, as well as those of Arabidopsis thaliana, for a better understanding of DNA repair mechanisms and possible additional biological functions of this protein family. This is our pipeline, which you can find in the abstract and in the poster. This is the phylogenetic tree of MSH orthologous protein constructed with the neighbor joining method. Here you can see how each protein is grouped according to its corresponding type. All analyses were made for the 24 proteins. Here you can see the domain composition of MSH6 for the different studied species. Here there are 3D models by homology using Swiss model for MSH and 3. And here for MSH 6 and 7. MSH proteins were modeled three-dimensionally with Swiss model, FAIR2, and Robetta. The models were refined with mod refined. We did Ramanchandram plots to validate 3D models. Here are some statistics of the validation of refined models of MSH proteins. Interaptome analyses were performed with all proteins MSH2, 3, 6, and 7, with the species available in strain database. This is a co-regulated genes analysis using Genevestigator. These were made for all proteins from Arabidopsis. We found using in silico protein-protein interaction and co-regulated genes analysis of orthologous MSH protein using strain database and Genevestigator respectively that MSH proteins is related not only to DNA repair function, but also to genes involved in cell cycle regulation, embryonic development, and mitotic or meiotic recombination. Functions that are conserved in Arabidopsis, rice, corn, and soybean. Therefore, it could be interesting to carry out future experimental studies to elucidate these possible functions. Thank you.